The most important point that came out of this study, though, was this single mutation in 3,000 cells rendered the animals diabetic. You see here blood glucose levels after a glucose tolerance test in normal animals and in the transgenic animals. And you can see here the dramatic difference in the glucose excursion that we create. So somehow glucose sensing in this small number of neurons is protecting the animals against diabetes. And when we block that, the animals become diabetic. I point out that these are young, lean animals. They're not high-fat, fed animals. So this is something that's happening at baseline. And our suspicion is that this nucleus is somehow mediating a neural suppression of hepatic glucose production. And when that suppression is lost, we get increased glucose release and therefore increased glucose excursions during a glucose tolerance test. This is all very interesting, but the important point is exactly the same effect happens in obesity. Obese mice have melanocortin neurons that don't sense glucose, and we think this might be one of the bases by which obesity induces diabetes. What you see here is curves of a control group, lean group, and a high-fat fed group. And you can see here, as I showed you before, glucose is increasing MSH secretion, whereas in the fat animals it's causing no effect. And this is just a further analysis of that data set. Now, a similar thing happens in the beta cell in diabetes, and it's probably due to a molecule called uncoupling protein 2. In the beta cell, uncoupling protein 2 sits across the mitochondrial membrane, and it lets protons leak out of the mitochondria. And that means that as glucose undergoes oxidative phosphorylation, there is no proton gradient to drive the production of ATP. And so in the beta cell, this means that you get a failure of glucose to stimulate insulin secretion. And so we investigated whether uncoupling protein 2 was present in the brains of diabetic mice. And indeed, that's what we find here. In the brains of obese, glucose-resistant mice, there are high levels of uncoupling protein 2. This is an in situ plot showing the arcuate nucleus, the place we've been recording electrically from, in the base of the brain. This is the third ventricle, and the pituitary would sit down here. And you can see on a high-fat diet, wild-type animals have about a 50% increase in uncoupling protein 2 in this part of the brain. And by single-cell level, we see a dramatic increase in the level of uncoupling protein 2 in these POMC neurons. This OB is a model of mice that do not make leptin, and you can see in these leptin-deficient animals, we see a dramatic increase in the level of uncoupling protein 2 in this part of the brain. We also found that we can inhibit uncoupling protein 2, and my collaborator Brad Lowell had shown that there's a molecule called genopin that they isolated from a Chinese traditional medicine for diabetes. Genopin is a potent uncoupling protein 2 inhibitor, and what you see here in this plot is alpha-MSH secretion in response to an increase in glucose levels, and we're getting that increased MSH secretion I've shown you many times. In lean animals, genopin doesn't change that. In obese animals, in this plot here, you see that glucose normally stimulates MSH secretion. It's lost in the animals on a high-fat diet. But pre-treatment for 15 minutes with genopin restored that glucose sensitivity in these neurons. And in recent studies conducted since we got back to Australia, we show that in lean animals, injection of genopin into the brain has no effect on a glucose tolerance test. These animals have essentially normal glucose control. But in obese mice, when we inject vehicle into the brain, we get a glucose excursion equivalent to what you get in an obese animal. Pre-injection of genopin, an hour beforehand, reversed that, and we get a glucose excursion curve like a lean animal. So 1 hour pre-treatment of the brains of these animals with genopin is reversing diabetes. Now the question, of course, can genopin be developed into therapeutic for humans? I think this is still an open question. There's good reason to suggest that genopin has a safety and tolerability history that probably makes it unsuitable. But nonetheless, I think it's a useful pharmacophore upon which to model potential future therapies. So in summary, POMC neurons sense glucose via a beta cell-like mechanism, and POMC neuron glucose sensing is necessary for normal glycemic control. This is critical, I think, because it highlights a new circuit in the brain that we can test for diabetic therapies. And it's potentially a new mechanism by which normal glucose homeostasis occurs. Uncoupling protein 2 is increased in these neurons of obese animals, and inhibition of that process restores normal glycemic control to these animals. Obviously, there's many questions this work relates, and particularly with relevance to this meeting. It shows the convergence of many signals onto these melanocortin circuits. And I think the fact that we can show parallel effects of, for example, leptin, GLP-1 and PYY raises the question of combination therapies. Combination therapy is the standard of care in many therapeutic practices in chronic disease. And so I think the idea that we can show individual effects on this neuronal circuit, and we know this circuit is important, really paves the way for testing what many drugs in parallel do. I think an obvious question is, does this melanocortin circuit transduce some of the effects of gastric bypass surgery? And in particular, I think studies to investigate what happens when you perform some of these procedures on mice that lack intact melanocortin circuits are going to be particularly interesting. There's obviously humans out there who also have deficient melanocortin signaling. I think it's a common question in the field whether part of the superior efficacy of gastric bypass surgery over pharmacotherapy is because pharmacotherapy might alter the levels of individual molecules from the L cell, for example, like PYY or GLP-1, whereas with bypass you have the potential to activate the L cells and lead to the release of many of the L cell constituents and also other cells in the region. And I think studies that really address these hormonal levels during and after bypass are going to be critical. And finally, what is the place for the combination of surgery and pharmacotherapy? In particular, my friends in Melbourne, Paul O'Brien, John Dixon and Wendy Brown, have talked about how banding surgery leads to similar 2-year efficacy to bypass, but how it takes about 2 years to reach that level of efficacy. What happens if in that window between the weight loss associated with banding, you aggressively treat with pharmacotherapy? Do you achieve similar endpoints more rapidly? And again, I think combination therapy is the standard of care in chronic diseases. I'm putting together a symposium at a meeting in Oregon in about a month or so, and we looked around for studies combining surgery and pharmacotherapy, and we couldn't find very much at all. And so I think the way is pay for some of the studies there. So in conclusion, leptin and glucose need the melanocortin circuits to produce their effects on energy balance, and we've shown that molecules like PYY and GLP-1 also act through this circuit. Diet-induced obesity causes a loss of sensing of glucose and leptin in melanocortin circuits, but in related work we've shown that PYY still acts in these circuits of obese animals. And so I think it suggests that this might still be the pathway through which therapies that activate the gut might be causing weight loss. This work does not address how dietary fat causes leptin resistance in the melanocortin circuits, and I think there's some good data suggesting this might be an inflammatory process. Obviously, resistance to leptin and glucose in these circuits is not the cause of obesity, it's a consequence of obesity. And I think when we start to ask questions about why people become obese, we need to look elsewhere in the brain. And in particular, I think we need to look at the reward circuits, because it's there that I think people are making bad decisions about food and overriding the homeostatic inputs from this part of the brain. The final question the lab's very interested in is how does this resistance to leptin and glucose cause the metabolic syndrome? And we're investigating what the impacts of leptin and glucose resistance in this part of the brain are on hepatic glucose production, skeletal muscle glucose uptake, as well as how leptin might be contributing to hypertension and tachycardia. And in related work with pharmaceutical companies, we have interest in how melanocortin activators like serotonin, dopamine and UCP2 molecules can have a practice in therapy development. I owe thanks to my funding sources and my staff and my collaborators and the audience. Thank you very much.